Welcome to our lecture on the fundamentals of biomolecules. Today, we are going to explore a topic that is literally the foundation of every living cell in your body: saturated and unsaturated fatty acids. We will discover how microscopic changes in a molecule's shape can determine whether a fat is a solid block of butter or a flowing liquid oil, and why this simple difference is a matter of life and death for our cells. Let us start by building some intuition. Imagine you are trying to pack a box full of straight, perfectly smooth wooden planks. Because they are straight, you can stack them tightly together, leaving almost no empty space, which creates a dense and solid block. Now, imagine trying to pack a box with tree branches that have sharp bends or kinks in them. No matter how hard you try, the bent branches will push against each other, leaving lots of empty space and creating a loose, flexible pile. This exact same principle applies to the fats in our food and our bodies. Now that we have a mental picture, let us look at the formal biochemistry. A fatty acid is simply a long chain of carbon atoms attached to hydrogen atoms, with an acidic carboxyl group at one end. When we say a fatty acid is saturated, we mean that every single carbon atom in the chain is holding as many hydrogen atoms as physically possible. There are only single bonds between the carbon atoms, which keeps the entire molecule straight and rigid. Just like those straight wooden planks, they pack tightly and form solids at room temperature, like butter. Building on that definition, what happens when we change the bonds? An unsaturated fatty acid contains one or more double bonds between its carbon atoms. Because carbon can only form 4 bonds total, creating a double bond means the carbons must let go of some hydrogen atoms. The molecule is no longer saturated with hydrogens. In nature, these double bonds usually occur in a specific geometry that forces the carbon chain to bend sharply at that exact spot, creating a permanent kink in the molecule, much like our bent tree branches. Why does this microscopic shape difference matter so much? Understanding the difference between these two types of fatty acids is absolutely crucial for understanding how living cells survive. Every cell in your body is surrounded by a protective cell membrane, made mostly of these fatty acids. If your cell membranes were made entirely of saturated fats, they would pack together so tightly that the membrane would freeze solid, preventing nutrients from entering or waste from leaving. By mixing in unsaturated fatty acids with their bent shapes, the cell membrane remains fluid, flexible, and dynamic. Furthermore, in our diets, consuming too many saturated fats can lead to tightly packed plaques in our blood vessels. To study these molecules efficiently, we need a universal language. Biochemists use a specific shorthand notation to describe the exact structure of any fatty acid without having to draw the whole molecule. The formula is C:(D). Here, C represents the total number of carbon atoms in the entire fatty acid chain, including the carbon in the carboxyl group. The symbol D represents the total number of double bonds present in the molecule. If D is 0, the fatty acid is completely saturated. When the number of double bonds increases, the molecule becomes more unsaturated, which drastically lowers its melting point and makes it more fluid. Now, let us look at the mathematical relationship for the tail of a saturated fat. For a fully saturated fatty acid, the general chemical formula for the hydrocarbon tail is CnH2n plus 1. In this formula, N is the number of carbon atoms specifically in the tail, excluding the carboxyl head group. The expression 2n plus 1 gives us the exact number of hydrogen atoms attached to that tail. As the carbon chain length N increases, the number of hydrogens increases proportionally, maintaining that straight, tightly packed structure that defines a saturated fat. Let us apply this formula to a real-world example. A student is given a bottle of palmitic acid, which is labeled with the biochemical shorthand 16:0. We are asked to find the total number of hydrogen atoms in the aliphatic chain. First, we read the notation: 16 total carbons and 0 double bonds. Because there are 0 double bonds, it is fully saturated. We subtract 1 from the total 16 carbons because 1 carbon is permanently part of the carboxyl group. This leaves exactly 15 carbons for the tail. Plugging 15 into our formula, we multiply by 2 and add 1. This reveals there are exactly 31 hydrogen atoms attached to the carbon tail. Now let us prove what physical state this molecule will take based on our calculation. We found the tail is C15H31. By adding the carboxyl group back, the final complete chemical formula is C15H31COOH. Because it holds the maximum possible number of hydrogens, we can confidently state that palmitic acid is a straight, fully saturated chain. There are no double bonds to create bends. This straight shape means the molecules will pack tightly together, just like the wooden planks we discussed earlier. Therefore, we have proven that palmitic acid will likely be a solid at room temperature. Moving on to unsaturated fats, the basic notation isn't enough. We need to know exactly where the kinks are. Biochemists expand the notation to C:D, followed by the Greek letter delta, with superscript numbers X and Y. The delta symbol indicates the position of the double bonds. The superscript numbers X and Y tell us exactly which carbon atoms start the double bonds, counting from the carboxyl end. If a molecule has multiple double bonds, we list all their starting positions. Each double bond represents a sharp bend in the molecule, so knowing these positions tells us exactly how the molecule is shaped. Let us decode a complex unsaturated fat. You are analyzing a sample of linoleic acid, an essential fatty acid found in plant oils. The bottle is labeled 18:2, delta 9,12. We need to identify its structural features. Breaking this down, the number 18 tells us there are exactly 18 carbon atoms in the entire molecule. The number 2 tells us there are 2 double bonds present, classifying it as a polyunsaturated fatty acid. Looking at the superscripts, the first double bond starts at carbon number 9, and the second starts at carbon number 12. This gives us a complete mental picture of an 18-carbon chain with 2 specific kinks. Let us logically derive the physical state of linoleic acid from these structural features. We established that there are 2 double bonds at positions 9 and 12. Because carbon geometry changes at a double bond, this molecule has 2 sharp kinks or bends in its physical structure. These kinks act exactly like the bent branches from our earlier analogy. They physically push neighboring molecules away, preventing them from packing closely together. Because they cannot pack tightly, they cannot form a solid structure. This loose packing is exactly why we can conclude that linoleic acid is a free-flowing liquid oil at room temperature. Let us bring all of this together visually. On the screen, you are looking at a direct comparison of these 2 molecular structures. On the left, notice the saturated fatty acid. It is a perfectly straight zigzag line. Below it, you can see how these straight chains stack tightly on top of one another, creating a dense, solid arrangement. Now look at the right side. The unsaturated fatty acid has a distinct sharp bend where the double bond is located, labeled as a kink. Below it, observe how these bent molecules interact. They cannot fit neatly together, resulting in large gaps and loose packing. This diagram perfectly illustrates why butter is solid and olive oil is liquid. We have reached the end of our journey, so let us summarize the key takeaways. First, remember that the physical properties of fatty acids are entirely dictated by their chemical bonds. Saturated fatty acids have only single bonds, creating straight chains that pack tightly into solid fats. In contrast, unsaturated fatty acids contain one or more double bonds, creating bent chains that cannot pack tightly, resulting in liquid oils. Finally, this simple difference in microscopic shape is incredibly powerful, allowing living organisms to fine-tune the flexibility and fluidity of their cell membranes to survive. You now have a solid understanding of saturated and unsaturated fatty acids.